Hi, welcome to the playlist on ketone bodies. This is actually a really exciting playlist because ketone bodies are these three structures that are shown up here. Number one on the left, this is acetone. In the middle here is acetoacetate, and on the right is beta hydroxybutyrate. These three molecules are called ketone bodies. Now, before we really get into what the function of ketone bodies are and why they're important, I just want to mention this, that if you look at the first two ketone bodies that are shown here, okay, you can certainly identify the ketone group. In the case of acetone, that's the simplest ketone you can have. And then in acetoacetate, there's the ketone right there with a carboxyl group attached to one of the carbons. Okay, beta hydroxybutyrate is not technically a ketone body, okay, because there's actually no ketone in this molecule. If you look here at this carbon, there's a hydroxyl group, and then over here there's a carboxylate. So technically, beta hydroxybutyrate is not a ketone body because it does not contain a ketone. But it turns out that acetoacetate and beta hydroxybutyrate are in equilibrium through a particular enzyme that we'll talk about in another video. Okay, but these three molecules right here are of extreme importance in producing energy in certain cases, and then also on top of that, it's just a really fun topic. Okay, now I want to start off this video by telling you something really important. First of all, there's many videos in this playlist on ketone bodies, and if you're really interested in reactions and mechanism, you might as well skip to the next few videos because that's where we'll do that. But what I want to tell you in this video is going to be more focused on, in general, what are the functions of ketone bodies and what's going on in the body that we would need to produce them. Okay, because in general, we have this big idea that ketone bodies are for starvation. They're used when people are suffering from, ketoacidosis, from diabetes mellitus. They sort of have a bum rap. Nobody likes to be starved. No one likes that. No one wants diabetes. So they have this bum rap about them. Nobody really seems to like ketone bodies, even though they are so important, especially in carnivores. In fact, carnivores, who are animals that eat nothing but meat, derive a tremendous amount of energy from ketone bodies. And we'll go into all sorts of stuff like that. And what I hope to do in this video not only is educate you about ketone bodies and their function, but I also want to dispel some myths. So I'm technically going to be acting as a myth buster here. And one of the myths that we're going to talk about is how ketone bodies don't give you energy. That your brain can't run on ketone bodies. And that's certainly not the case. And we'll go into why that is later in this video. And in the next few videos, you'll see that ketone bodies actually produce a tremendous amount of energy. And we'll get into that later on. But now that we're in the video, right, I want to tell you that this is going to be the general structure of the video right here. We're going to talk about ketone bodies, okay? And then we're going to talk about the two main diets, two really critical diets that would elicit the use of and production of ketone bodies. We're going to talk about essential versus non-essential amino acids. We're going to talk about beta oxidation, and then how you convert the products of beta oxidation to ketone bodies. And then lastly, we'll talk about their general function. And in the next few videos, we'll talk about the pathways and so forth, and mechanism and so forth. Okay? So at the very beginning of this video, we talked about the structure of the ketone bodies and how beta hydroxybutyrate technically is not a ketone body, as it possesses a hydroxyl group instead of the ketone. But since beta hydroxybutyrate can interconvert with acetoacetate through beta hydroxybutyrate dehydrogenase, we sort of group it with these. And in fact, that enzyme is very active in humans, and it does interconvert between these two. Okay, so by that logic, we term the three ketone bodies acetone, acetoacetate, and beta hydroxybutyrate. Now, acetone at one time was thought to be just sort of a side product. In fact, what ends up happening, and I'll do this in yellow, is what can happen spontaneously in humans is that you can split this bond. Okay? And it happens non-enzymatically. When I say spontaneous, In this context, I mean that it's non-enzymatic, okay, or at least it's thought to be. We know the activity exists, but acetoacetate can spontaneously decarboxylate in solution, and you end up generating this guy, which is acetone. And for a while, acetone was thought to be sort of a static molecule. In fact, in somebody with diabetes, the smell that's on their breath when they're suffering from ketoacidosis is acetone. So it was thought to be exhaled, excreted, and so forth, but we now know it to have a very bizarre metabolic pathway for its catabolism. And what you would see in the acetone video is that it produces a tremendous amount of energy for something that's only 3 carbons. I mean, you look at this molecule, really small, it's the simplest ketone that you can have, but yet it produces so much energy, and that's very bizarre. But in fact, it does. Okay, so I hope you understand the structures here. Acetone is the simplest ketone. Acetoacetate is basically the simplest ketone, but with an added carboxylate group on it. And then beta-hydroxybutyrate is just the reduced form of acetoacetate in terms of the ketone. Okay, it's been reduced into a hydroxyl group. So these 3 guys right here, these are the ketone bodies. Okay, now when would ketone bodies become important? Well, I'm going to give you a brief overview of when they would become important, but once we do that, you sort of have to know a little bit of background about what's going on in the body that would lead to their production. Because I can't just straight up tell you this is when it happens. There's a certain way you have to teach ketone bodies, and I hope to elucidate it here. So there's 2 main diets that would be large candidates for producing ketone bodies. Okay, one of them is a starvation mode. So this is the fasting state. Okay, let's imagine for a second that, maybe at one point in the day, you ate a huge carbohydrate meal. Maybe it had some protein, maybe it had some lipids, but the idea is that it had carbohydrate. Okay, and assuming, let's say it's a potato. Okay, you had like a steak and a potato. So obviously the potato has plenty of carbs, plenty of things that degrade to sugars in your duodenum. They get degraded down to that, and they get absorbed into the blood. And ultimately what ends up happening is cells intake them, particularly the liver and the muscle, and they eventually burn some of it in glycolysis. But the rest of it that's not used for that purpose, once the enzymes in the TCA cycle are allosterically inhibited, the glucose ends up getting phosphorylated by hexokinase. Phosphoglucomutase converts it to glucose 1-phosphate, and then you get an enzymatic reaction of glycogen synthase that produces a glycogen polymer. And what glycogen is, if you haven't seen it, is glycogen is a storage form of glucose. It's a polymer of glucose molecules. Okay? So you have this glycogen polymer that's basically a storage form of glucose. Okay? And the main places that you find glycogen are going to be in the skeletal muscle and the liver. Okay? Now, let's imagine for a second that this happened like 4 hours ago. So at this point, you haven't eaten anything, and that's the assumption we're going to make. Is we're going to make the assumption that that meal was there, but now we're sort of going off of that meal, and we haven't eaten anything since. Well, eventually what's going to happen is you're going to start going into the fasting state. And so what's going to happen is you're going to burn that glycogen. In other words, there's an enzyme called glycogen phosphorylase. And glycogen phosphorylase is going to break up the glycogen polymer into glucose 1-phosphate molecules. The glucose 1-phosphate will go into the enzyme phosphoglucomutase, and by Le Chatelier's principle, it'll force the production of glucose 6-phosphate, which goes into glycolysis. And so by that logic, what ends up happening is you're burning the glucose. Okay? But there comes a point at which you run out of glycogen for the most part. Okay? If you're running out of glycogen, you have to find a way to produce ATP. There's no other option. You've got to produce ATP. So if you've run out of glycogen, your blood sugar's low, you've got to find another way to get your blood glucose to normal levels. So that's number one. You got to get your blood glucose to normal levels. And number two, you've got to find other ways to produce lots of ATP, not just for your liver, but for the rest of your cells, especially your skeletal muscles and your brain. And it turns out that the way you do that is through ketone bodies. That's one of the ways that you can do it. So in the starvation mode, that's one area that you'll produce ketone bodies. Don't worry, we're going to go in more detail on that in a few minutes. But I just wanted to mention at this point that when you're in the starvation mode, what ends up happening is you're going to be producing ketone bodies. And what I will say at this point is that the liver is what does that. Okay? This is one reason you cannot live without a liver. Okay? Sure, the liver produces, complement enzymes, coagulation enzymes, and along with the fact that it detoxifies foreign compounds, it does this. It produces ketone bodies. It's the main performer of gluconeogenesis. So it's an incredibly important metabolic organ. And so that's one of the many, many, many reasons you can't live without it. Okay, so it's really important here in this video and in its physiology. Okay, the other main diet that will produce ketone bodies is, let's say you are in the fed state. And by the way, in the fed state, we sort of have this idea that it's, you ate a carbohydrate-rich meal. But that's not necessarily the case. There are some people, including myself, who take pride in eating a very, very high protein and lipid diet and an extremely low carbohydrate diet. So basically we're running on basically nothing but meat. And I go back to this experience I had before I was a biochemist. And before I was a biochemist, I was in a nutrition class that was taught in a kinesiology department. And by the way, this teacher taught all sorts of incorrect things. And I distinctly remember there was a guy in there. We had this assignment basically. And the assignment was we had to, give a short 5-minute presentation on some, fad type of diet. Some area of nutrition that interested us and so forth. And the very first guy that came out there was somebody who was advertising the paleo diet. And if you don't know what the paleo diet is, it's something that eliminates dairy, it eliminates breads, and basically what your diet boils down to is it's meat, vegetables, a little bit of fruit, nuts, seeds, and so forth. Okay? But what this teacher purported was that you had to get like 60% of your calories from sugar. And by the way, that's not good to do, number one. But I distinctly remember him coming up and giving this presentation on the paleo diet, and her comment was, how the heck do you have energy? How the heck do you have energy? Well, imagine what this guy is doing, and what I do, is we're intaking a very, very high protein and lipid, which in other words, lipid, by that I mean triacylglycerol, diet. Well, obviously I can function. I'm making this video. I work out like 3 hours a day. So there has to be some way you get energy. Well, it turns out that what happens is, again, your liver plays a huge role in this, is that the lipids that I obtain from my diet, first of all, the duodenum hydrolyzes them down to fatty acids. They get intaken through chylomicrons, and then the liver processes them. And through this process, ketone bodies are made, and they're basically dumped into the blood, and they go to peripheral tissues and serve them. So there's a huge distinction, though, between a starvation or fasting state ketone body production and a fed state ketone body production. In the starving state or the fasting state, the lipids that you're turning into ketone bodies, those lipids come from your adipocytes. If you don't know this, adipocytes are your fat cells. So like if you have gut fat, or butt fat, whatever it is, that's where those fatty acids come from. They're mobilized, particularly by the action of glucagon. They're mobilized, they go to the liver, bound to serum albumin, so they get to the liver, they're intaken by the hepatocytes, and then from there, those lipids get turned into ketone bodies. Okay, that's a really critical thing to understand. They get turned into ketone bodies, and then they get dumped into the blood. But if you're getting it through the diet, so you're in the fed state filled with amino acids and lipids, then you don't have to mobilize those things from your adipocytes. They instead come directly from from your duodenum. They come directly from there, and then you basically take them into the liver cells, and then they're converted into ketone bodies. Okay, so there's two basically different mechanisms that you use to make these guys. Okay, but in any case, the liver makes them, dumps them into the blood. They travel through the blood, go to peripheral cells like the muscle, the brain. They're intaken into those cells and then used. Okay, so I just want to get this thing out of the way that there's two primary diets where you'd be doing a lot of this. Is if you're getting a lot of fat from the diet, a lot of protein from the diet. Now the protein is not usually converted to ketone bodies. It can be. We'll talk more about that in just a minute, but there's certain conditions under which that occurs. Okay, but in general, your lipids are what get converted to ketone bodies. I just want to stress the fact that there's two different mechanisms by which that occurs. One of them, which is the fasting state, it comes from adipocytes. In the case of the fed state, it'll come from lipids obtained from the diet. Okay, so now we've got this data, this information out of the way. We need to talk about what's going on in the liver, because the liver is really the guy who's doing everything. So let me sort of draw a depiction of the liver. So this right here, this is your liver. Not very good picture, okay? But basically what we're going to talk about is essential versus non-essential amino acids. Let's define that first. An essential amino acid is an amino acid that we do not possess the enzymes to make. Okay, so if we don't have the enzymes to biosynthesize it, then you have to get it through the diet. There's no other option. So essential amino acids, if you're not getting it through the diet, you have a deficiency therefore. For example, tryptophan. That's an essential amino acid. We don't have the enzymes to make tryptophan. Phenylalanine, tyrosine, these are also essentials. We don't have the enzymes to make them. Histidine, of course, I'm naming all the aromatics right now, but there's other ones. Valine, leucine, isoleucine. We don't have the enzymes to make those guys, so we have to get them through the diet. But there's also non-essential amino acids. These are amino acids that we actually have the enzymes to make them. Okay? We have the enzymes to make these guys, and so if we have the enzymes to make these guys, then we don't have the absolute requirement of getting them through the diet. Okay, we can get them through the diet, and that's more of a supplementation, but we do have the enzymes needed to make them. Okay, so I want to get that out of the way, essential versus non-essential amino acids. Now what I'm going to do is, I'm going to do this in a different color. Okay, I'm going to draw another liver here. This one's in red, the other one's sort of in maroon. What I want to do is I want to talk about when you'd be degrading essentials and when you'd be degrading non-essentials. Okay, so in this liver right here, this liver is going to be my fasting liver, and this is going to be my fed liver. So just a distinction between the two. Fasting is when I'm starving, okay? The fed liver is going to be when I have all those, lipids and amino acids from the diet, okay? So let's talk about the fasting first, okay? Let me ask you a question. Between essentials and non-essentials, which is the one that you would primarily like to catabolize? And if you said non-essentials, you're right. The reason you'd like to catabolize non-essentials over essentials is because essentials you can't make. They're usually the rarer amino acids. You can't make them. So if you can't make them, it wouldn't make sense for those to be the ones that you degrade. You would preferentially like to degrade the non-essentials because you can make those. So usually what happens in the fasting state is you get proteins, which I'll designate as a P. The proteins will get degraded into, because if you hydrolyze proteins, they get hydrolyzed into the essential amino acids plus the non-essentials. Now the essentials are essentially held onto, but the non-essentials are in general what get degraded. Okay? Now it turns out that what the non-essentials get degraded to, so this is enzymatic, I'll put ENZ for enzymatic. Through enzymes, the non-essentials get degraded to either pyruvate or TCA cycle intermediates. Okay, these are the two main things. Let me do this in light blue. These are the two main things that the non-essential amino acids get degraded to. Okay, the essentials are sort of held onto, but in general, non-essential amino acids tend to get degraded to pyruvate or TCA cycle intermediates. The essentials actually tend to get degraded to some TCA cycle intermediates, but in general, the essentials get degraded into things like acetyl-CoA and so forth, and acetoacetyl-CoA and things like that. But we're not really degrading the essentials right now. Those are being held onto. What we've essentially done is activated the enzymes that degrade non-essentials, and we get pyruvate and TCA cycle intermediates. Now, we won't go into the nitty-gritty details here of what happens, but essentially these guys, through gluconeogenesis, get converted into glucose, and the glucose gets dumped out of the liver into the blood. So now the glucose is in the blood, and it goes to serve peripheral tissues. Okay, this process right here, this is called gluconeogenesis. The process of protein getting degraded to the essentials and non-essentials, that's the process of proteolysis. Okay, the lysis of proteins. The conversion of non-essentials to pyruvate and TCA cycle intermediates, that's just non-essential amino acid catabolism. And then the process of converting pyruvate and TCA cycle intermediates into glucose, that's the process of gluconeogenesis, and then glucose gets dumped out of the liver into the blood. That goes to serve peripheral tissues like the muscle, make sure that's an M, a muscle, the brain, and so forth. Okay? But this is in the fasting state, because you don't really want to degrade essential amino acids. What you tend to degrade are non-essential amino acids. But that is a different case than in the fed state. So now we're talking about the fed state. Now let me draw the intestine right here. So let's pretend this is the intestine. Okay. In the intestine, I'll do this in a different color just so it doesn't get monotonous. We have a protein. And the protein in the intestine, and also the stomach, because remember, we have things like pepsin in the stomach, but there's also intestinal proteases. They convert these proteins into amino acids. Okay? The amino acids then get into the blood, so they traverse into the blood, and then the amino acids will end up in the liver. Okay? And this is in the fed state, and these amino acids are comprised by both essentials plus non-essentials. Okay? But here is the big difference between the fasting state and the fed state. In the fasting state, we're getting these proteins basically from the liver. Okay, the liver has lots of proteins, and if it needs to make glucose, it will just degrade these proteins into essentials and non-essentials. And of course, there's probably some amino acids just floating around in any case. But it will degrade proteins from its own cells. But see, in the fed state, those proteins get degraded to amino acids in the intestine, and those get transported into the liver, where you now have essentials plus non-essentials, and we're actually going to degrade both of them this time. And here's sort of the way to conceptualize it. Amino acid levels in the blood and in cells like to be tightly controlled. So the way that you control an amino acid's concentration is by its synthesis and by its destruction. In other words, by its biosynthesis and by its catabolism. So if these amino acids were to build up to really high levels, you've got a problem. In fact, for example, high levels of cysteine cause something called acute cysteine toxicity. So you don't want extremely high levels of an amino acid, so the way you get around that is you catabolize it. Now here, the story is the same for non-essentials. You're just going to have an enzymatic degradation into pyruvate, TCA cycle intermediates. Let me again circle that in blue. In any case, what's going to happen, assuming you're eating a high protein and high lipid diet, is this is going to get converted into glucose, and that's of course going to get dumped into the blood and go and serve peripheral tissues. But the story of essentials here is a little bit different because we have to really tightly control those levels. So what ends up happening is the essentials are actually going to get degraded enzymatically, so ENZ for enzymatic, and they tend to get degraded into two things. These are in general what they tend to get degraded to. Number one, they can get degraded into a TCA cycle intermediate, which is in general succinyl-CoA. There are some amino acids that get degraded to succinyl-CoA, which is a TCA cycle intermediate. But the other things they tend to get degraded to are acetyl-CoA, which I've drawn there, and I'll kind of have to come out here to do this. They get degraded to something called acetoacetyl-CoA. So these are the two things that essentials tend to get degraded to. Acetyl-CoA here and acetoacetyl-CoA there. And what ends up happening with these guys, let me circle these in dark blue. So these guys, they're still in the liver. And what ends up happening with those guys is they will get converted to ketone bodies. So this is really where we're coming back to this. These guys get converted to ketone bodies, and then these ketone bodies will get dumped into the blood, and they will go and serve peripheral tissues. So this is basically the amino acid story for both the fasting and the fed state. Okay, but what I want to do now that we kind of have an understanding of amino acids, is now go to the lipid component. Because all of this stuff, this is just amino acid stuff. But we need to really get to the lipid stuff. So I'm going to come down here and do this. And what I'm essentially going to do is I'm going to just draw one liver this time. I'll go back to doing it. Actually, let me do a different color. Let me do orange, just so we know it's different. So this is really going to be the case for both fasting and fed state, because in general the physiology is the same. But what I want to do is this. Here I'm going to have fasting. This is the fasting case. And this is going to be the fed case. So in the fasting state, what we mentioned is that the lipids are coming from adipocytes. So here we have oops, didn't spell that right. We have an adipocyte, which is going to release fatty acids. Those travel through the blood, and they end up in the liver. In the fed state, you have your intestine, right? So this is your intestine. And you start up with triacylglycerol, but enzymatically that gets converted to fatty acids. And those, of course, get bound to chylomicrons, and they're going to end up in the liver. Okay, so the story is going to be the same in general for both of these cases. But what I want to have you remember is that for a fasting state, those fatty acids have to be mobilized from the adipocyte. In the case of the fed state, you're just getting it through the diet, so it has to exit through the intestine and end up in the liver. But in both cases, these guys are going to end up in the liver. Okay. Now here's the thing. These fatty acids are essentially going to end up being activated, and they get activated through an enzyme called fatty acyl-CoA synthetase. And so what you end up getting is something like this. So let's say I'm just going to draw a small one. But you have 1, 2, 3, 4, 5, 6. Let's say you have that. In any case, it'll end up being much longer than this. But this right here, this is basically a fatty acyl-CoA. Okay, this one happens to be 6 carbons. In general, they'll be much longer than this. But the idea is that the fatty acid will get activated into this guy. Okay. Now, what's essentially going to happen to this guy is it will go through beta oxidation. Now, in this case, it's 6 carbons, so what you would end up getting is 3 acetyl-CoA for this guy. But what I'm going to do is I'm going to say, okay, well, let's just say you get N. Let's say you get N acetyl-CoA. Okay, it'll be some number which will depend on how many carbons the fatty acyl-CoA is, but in general, it's just going to be some amount of acetyl-CoA. Okay, now what we're going to say is the acetyl-CoA will go into ketone body biosynthesis. Okay, so the story is really the same in both of these cases. Okay, the fasting state and the fed state. The fatty acids that get beta oxidized in the liver will essentially undergo beta oxidation, and they will end up getting turned into acetyl-CoA molecules, and that will go into ketone body biosynthesis. Of course, then the ketone bodies will be dumped into the blood, and they will serve peripheral tissues like the brain and muscle especially. Okay, so this is the story for fatty acids. Now, what I also want to mention at this point, because it is kind of important, is that per round of beta oxidation, let me do this just so you know it's still part of the liver. Per round of beta oxidation, you're going to get an NADH, and you're going to get a totally reduced ubiquinol. The NADH will serve NADH ubiquinone oxidoreductase, which is complex 1 of the mitochondrial respiratory chain. Coenzyme Q reduced or ubiquinol will serve complex 3, which is cytochrome C ubiquinol oxidoreductase. Each of these enzymes pumps protons when they're activated, and so the pumping of protons powers ATP synthase. So the way in these two states, when you have lipid and protein in the diet or you're in the fasting state, the way the liver gets its energy is through beta oxidation. It produces NADH and reduced ubiquinol, and those power proton pumping, which powers ATP synthase for the synthesis of ATP. Okay, so that's the general physiology of what's happening. Again, we're not going into mechanism here. That's saved for another video, but I hope this makes sense. Okay, you have amino acid oxidation or catabolism that's occurring in the liver, different ways in different states, but in general for lipids, the only thing that really differs for these two states is how the lipids get there. In the case of the fed state, it gets hydrolyzed in the intestine to fatty acids. Those get transported into the liver through chylomicrons. In the fasting state, the fatty acids come from the adipocyte. They bind to serum albumin, travel in the blood, and then go to the liver. Okay, but here in the liver, the story is the same. Okay, so beta oxidation, we've covered that. And conversion to ketone bodies, the only thing I'll mention here is just that the acetyl-CoA that's produced, down here from beta oxidation, that gets converted to ketone bodies. Okay, we have a whole video on this conversion, and I recommend that you go watch it. But there's a whole video on the conversion from acetyl-CoA to ketone bodies, and it's a series of, as I remember, about 3 or 4 enzymes that does it. Okay, but the ketone bodies that are made, those are these guys up here at the top: acetone, acetoacetate, and beta hydroxybutyrate. They get dumped into the blood. They go and serve peripheral tissues. So that leads us to the final thing in this video: is what are the function of ketone bodies. In part, you'll have to see the pathways of the ketone bodies to really understand the fundamentals of their function. But the idea is that they are for producing energy. Okay, acetone especially produces a whopping amount of energy, and actually so do the other ones. Okay, so their function is energy production. Okay, so if you imagine what's happening with the liver is trying its best to maintain blood glucose levels. Okay, because blood glucose levels theoretically have to be maintained within a certain range. So the function of the liver is to maintain blood glucose levels by doing gluconeogenesis. The main way it does that is by degrading non-essential amino acids to pyruvate and TCA cycle intermediates. And then it'll also do that in the fed state when you have lipids and amino acids from your diet. Okay? But the idea is the liver's trying to maintain blood glucose levels. The glucose can be uptaken by the brain, it can be uptaken by the muscle, but the really important thing to understand is that glucose only gets you so far. Okay? A whopping amount of the energy that you actually get is from ketone bodies. And I just want to have you think about something really quickly. You can probably think of many organisms out there that eat nothing but meat. For example, if you have a lion that's in the wild, they eat nothing but meat. A cheetah, for example. A cheetah eats nothing but meat. They don't eat, the crap that the people in the United States eat. Okay? All that sugary stuff. They don't eat that. They just eat meat. So what their diet is mainly going to consist of is this state, which is the fasting state, And the intestine, or not the intestine, the fed state right here. So the fed state here and the fasting state here. Those are the two main situations that a carnivore would experience. And if you are a carnivorous human, you will experience the exact same thing. But if you think about it, there's lots of, like, I wouldn't call it a conspiracy, that's not the right word, but there's all sorts of thought out there that you can't function on ketone bodies. Well, it's almost obvious that that can't be the case. If you think about a cheetah, they literally run 70 miles per hour. I think I read that, and I heard that they run 70 miles per hour, and they're chasing gazelles and all sorts of stuff. Do you really think that they could run on that if ketone bodies didn't work? Could something run like that if ketone bodies didn't work? Could a human who eats this way, could they exercise 3 to 4 hours a day if these ketone bodies didn't work? Ask yourself those questions. These ketone bodies are a tremendous source of energy, and I really hope to instill that in you in this video and the videos to come. One of the next videos is on acetone and acetone metabolism. Acetone produces a lot of energy, and we're only recently discovering that it has a really unique pathway, but that it does produce quite a bit of energy. Acetone will eventually get degraded to acetyl-CoA, and we'll see that later on. Okay, so hopefully this video helped you get a basic understanding of the function of ketone bodies. What I want to instill in you is that the liver is so important in doing this. The liver makes ketone bodies. It makes them for the purpose of energy for the rest of the body. If you didn't have a liver, you would not produce ketone bodies. So I hope this video helped you. See you in the next video.